My name is Ibrahim Latunde Olukudano. I'm presenting my paper titled Distributed Kriging-Bootstrapped Deep Neural Network Model for Real-Time Seizure Detection from EEG Signals. I work together with my professors, Professor Saraju Mohanty and Professor Elias Kogianos. And you can see our email addresses here for any questions after this presentation. This is the outline of my talk. What is seizure? Firing neurons in the brain generate tiny electric signals for every action that takes place in the body. These signals act as triggers for the specific actions, but sometimes these signals are abnormally transmitted without any request, and such actions result into seizure. You can see a seizure patient here, lack of consciousness and lack of control. That is the aftermath of a seizure attack. Why seizure detection? Approximately 10% of the world population will have at least one experience of seizure in their lifetime. Considering the current population of the world, that is a whopping 800 million people that will experience seizure in their lifetime. That is why we need seizure detection. Also, people living with epilepsy experience a higher mortality rate of about 44% compared to the general population whose mortality rate is only 12.2%. Therefore, timely epileptic seizure detection is an important first step towards effectively managing the seizure disorder. You can see here, with seizure detection, there can be timely intervention and the seizure can easily be controlled. Without seizure detection, there can be fatal injury resulting into death. These are the types of seizure. These are the characteristics of EEG signals. EEG signals have high complexity. They have low intensity in the order of microvolts and a frequency range between 0.5 and 30 Hertz. This is an example of a typical EEG signal obtained from the brain of one of the epileptic seizure patients in the data set that we are used for this research. These are the various research problems in seizure detection existing in the literature. These are the challenges we faced in the course of this research. Collecting a custom data set due to the stringent regulations that are involved is very, very difficult. There are stringent regulations involved when collecting data from animals or human subjects. The same also applies to testing our models on humans and animal subjects directly. There are stringent regulations that will not allow us to do that. It is also difficult to estimate the level of noise in public data sets since the conditions of the environment in which they are collected are not actually known. These are the related research in seizure detection in the literature. These various research works in the literature have combined machine learning and electroencephalography recordings for their seizure detection. There are other works that did not use EEG for seizure detection. These are some of the examples of such works that did not use EEG for seizure detection. Also in the industry, work is ongoing to develop seizure detectors that can be implanted into the brain of the patient for fast detection of seizure. We can see here, IBM is developing an implantable seizure detector by leveraging on their neurosynaptic computing hardware called the True North. The True North has a size that is like the postage stamp and can be implanted into the brain of the patient. You can see this is actually the hardware representation of the True North, and it consumes very minimal power, that is 1,000 times less than a conventional processor of similar size. Also, there are consumer electronics devices in the market for seizure detection. You can see this now, Embrace 2, and this other device, they actually work based on the skin conductance level of the patient. When a patient is under a seizure attack, the conductance level of the skin is high. And so they set a particular threshold so that when the conductance level of the skin is above that threshold that have been set, then a seizure is flagged. So that is how this is working. What are the drawbacks of existing works? High seizure detection latency. The models are too complex and unrealistic for real-time deployment in the Internet of Medical Things, and of course, lack of adequate intervention mechanism after detection. So the typical detection latency in the literature is around 4 to 6 seconds. Early detection is about 1 to 2 seconds. Most works in the literature fall around this typical latency of detection, which will not be good enough for real-time Internet of Medical Things application. Research question. While the Kriging-Bootstrapped deep neural network seizure detection model that was prior to this work reduced the total training time by 75% compared to the ordinary deep neural network model. The major contributor to the reduction is DNN. The Kriging time remains unchanged. Is it therefore possible to achieve a further reduction in training time by distributing the Kriging computation across different cores without affecting the overall performance of the seizure detection model? These are the novel contributions of this work. It achieves a seizure detection latency of less than 1 second. A novel single channel seizure detection was also achieved, and then we achieved 91% reduction in training time and performance improvement by at least 2.5%. Kriging. Kriging was originally developed as a geostatistical model for spatial prediction. It is a stochastical process that is governed by a mean value and the relative covariances of known data points with respect to an unknown. You can see this figure. We have 3 locations with known values, and we have 1 location with an unknown value. Using Kriging, the value at the unknown location can be estimated. Why Kriging? Kriging has been used in this case because the brain can be modeled as a spatial map on which spatial data processing methods can be applied. And then Kriging has good performance on relatively small data sets. It also comes with an estimation variance that tells us how good we are doing with the prediction, and it uses few hyperparameters to achieve a very high accuracy. These are the existing applications of Kriging. Brain as a spatial map. The brain can be modeled as a spatial map, and in fact has been modeled as a spatial map with respect to this work, and you can compare it to the geographical map. You can see that as we have links and locations with different values on the geographical map, we also have the same thing represented on the brain, with the red dots representing the locations where there are seizures, and then the black dots representing locations of unknown status. So with Kriging, we can be able to detect the status of the unknown location if they have seizures or not. These are the mathematical expressions and derivations for Kriging estimates. The bootstrap. A bootstrap is a strap that is looped and sewn to the top of a boot for greater ease in pulling it on. It simply means getting oneself through difficulties only with the resources at one's disposal. So bootstrap sampling. The bootstrap method that we used for Kriging, the original term was borrowed from bootstrap sampling. Bootstrap sampling refers to a method of estimating the true statistical value of a population from some given samples. It involves sampling from the available samples with replacement to create multiple sets of samples. The number of sample sets that is created from the original samples is called the bootstrap size. So you can see now that you get these statistics from sets of samples, and then you can now summarize them, into the kind of statistical value you are trying to estimate. So we have adapted that to the concept of bootstrapped Kriging, that we, in such a way that we have an initial field with relatively small amount of data size, and then we bootstrap it by increasing the data size using Kriging without changing the field, the area of the field. So this is deep neural network, where we achieve a forward propagation to calculate the cost function, and then we use backward propagation to calculate the weights and biases so that we can get our final prediction using this formula here below. Now, the concept of distributed Kriging, is such that we distribute the Kriging computation across multiple cores. As you can see in this figure, we have cores 1, 2, 3 up to core n. So the Kriging computation are distributed in such a way that the computation happen concurrently, and then we can reduce time by doing that. So after we have done the concurrent computation, the outputs are aggregated and then passed into the deep neural network. So this shows the computational analysis of the distributed Kriging, and then this shows our proposed fast and accurate real-time seizure detection model. Now, this is the training and learning process for the distributed Kriging bootstrapped deep neural network seizure detection model. So we have an initial input, then we distribute them across different cores with a given bootstrap size B, and then we aggregate all the outputs, and then we pass them into the deep neural network to obtain the final status of the patient, if it's seizure or not seizure. So we have used various data sets. We use the Bond data set, popularly used in the seizure detection research community. And we have also used the MIT SCALP data set, which is also very, very popular in the seizure detection community. These are the various features that can be extracted from EEG signals, but the ones in red are the ones that we have used for this work. And these are the mathematical expressions. For Jot complexity, you have this formula, and then for singular value decomposition entropy, we have this formula, and for the Petrosian fractal dimension, we also have that formula there. So this is the DNN specification for our DNN architecture, and then this is the result. We can see that with the distributed Kriging-Bootstrapped DNN model, the time taken reduced. We were able to reduce the training time, for the DNN. I mean, for the distributed Kriging-Bootstrapped DNN model. It achieved a testing accuracy of 100% and a training accuracy of 99.92%. When compared to other methods, we could see that we achieved a reduction in training time by 91%. And then you can see the drastic fall in the training time here. And then you can see that the performances in other aspects were not compromised. And then you see that the detection latency was 0.80%. I mean 0.80 seconds. We also achieved a single channel seizure detection model, that is even more accurate than a multi-channel seizure detection model using the second data set. So when you compare the results, when we did not distribute the computation, we see that ordinary Kriging uses, consumes a lot of time. But in the distributed Kriging model, we see that, that it was almost a ratio 1-to-1 between DNN and Kriging. So when we compared our results to other works that use multiple channel, we see that our single channel achieves a better result. So in conclusion, we achieved a detection latency of 0.8 seconds, which is, novel. And then we also achieved, an improvement in training time by about 91%. So these are the future research for our work, and these are the references that we have used. Thank you.